Hello, I'm Doug Montgomery. I'd like to welcome you to our November Fireside Chat. I was just talking with our producer, Megan Gibson, and she's reminded me this is the 16th Fireside Chat. And look how time passes when you're having fun, because I always have a lot of fun doing these. For those of you who are maybe new to our Design of Experiments course, this Fireside Chat is a production that we run, that I host every month. I kid people that I'm channeling Franklin D. Roosevelt, who was, of course, president of the U.S. from 1933 to 1944. And he had pretty much monthly Fireside Chats too for that entire period of time. That's like 11 years. I'm not sure I'm going to be doing this for 11 years, but anyway, we always try to have a lot of fun. And part of the fun is I always have a guest speaker who talks about a specific problem or application or topics of design of experiments concepts. And we always have some time at the end of that for questions and answers. This is a great time for you to ask questions, and they can either be directed to me or to the guest. So you can ask about anything you'd like. You can ask about the course, questions that come up from Coursera, and we'll try to get all of those answered. If we don't get them answered during the Fireside Chat, we'll get them answered later on, and all of this will be posted in Coursera. Okay, quick reminder. Please keep your mics muted, your cameras off during the session. That'll minimize any sort of disturbances. And if you do have a question that you want to submit or something that just pops into your mind during our chat this morning, please do that using the chat function on your Zoom menu. And you don't need to wait until the Q&A starts to actually submit your question. You can do it anytime you want during the session. And as I said, if you don't get everything answered during this time, we will respond to it in Coursera for the people that are actually enrolled in the course. My guest today is Julia O'Neill, and she leads CMC Modeling and Statistics for Moderna and is a member of the Technical Development Leadership Team. She's a fellow of the American Society for Quality. She's provided consulting support for since 2005 on the development of many types of products involving things like gene therapy and regenerative medicine. She worked at Merck as senior scientific fellow, working in statistics and regulatory and analytical sciences, and director in global technical operations. And she had a big focus on biologics and vaccines. And her experience includes the development of specifications, stability analysis, validation of analytical methods, process development, design and implementation of continued process verification programs, and design of experiments for a wide variety of development activities in vaccines, biologics, gene therapies, small molecules, and many other products that are derived from biological materials. Julia is a Six Sigma Master Black Belt. She's managed and built a lot of teams and driven a lot of improvement efforts and solutions for problems that address regulatory issues. She's also provided encouragement for 5 colleagues to complete graduate degrees in statistics. She has a lot of years of experience bridging statistics at chemical engineering in the pharmaceutical and chemical industries. She has a master's in statistics from the University of Wisconsin and a BS in chemical engineering from the University of Maine. And I've worked with Julia on and off for a number of years and been involved in ASQ activities with her. And she is just an absolutely terrific person. And I think you're going to really enjoy her presentation. So Julia, you're up very shortly here.
Well, thank you, Doug. It's an honor to be invited to do this, and it's always a pleasure, of course, to talk about design of experiments and statistical topics generally with you. So let me see if I can share my screen to get started. here we go. Okay, and I'm hoping that that is coming through bright and clear for everyone.
Looks good to me.
Great. All right. So what I'm going to talk about today is the impact that design of experiments have had in vaccine development. And a little over a year ago, probably most of you would have, like my mother used to say, what's that company you work for? Ma. And of course, now Moderna has become a household word in a lot of households. But design of experiments actually played a big role in getting us ready to launch the vaccine for COVID, mRNA 1273, and have now many doses distributed. But I want to start by dedicating this to Connie Boer. Doug introduced me to Connie about 20 years ago. I was working in a chemical company, Roman Haas, and we were looking to bring in some training on robust process design. And Doug and Connie came in and delivered a great class on robust process design. And Connie and I really became friends over the years. I dedicate this to Connie because of I learned so much from her in many ways. She was a wonderful mentor and friend and statistics teacher, just a great person all around. But the thing I learned towards the end of Connie's life was that the importance of getting life-changing medicines and vaccines to people quickly. For Connie, her diagnosis with breast cancer until her death was less than a year. And at the time, I had just left Merck, where I was working on Keytruda, which is a breakthrough designation cancer treatment that had saved many lives, but we couldn't get Connie into a clinical trial for that or anything similar. And literally the clock ran out for her. And that was a real turning point for me where I said, boy, what good are medicines and vaccines if they can't get to the people who need them fast enough? So I really started to focus at that point on what does it take to accelerate the development and distribution of these life-changing products. So in 2021 now, we're 5 years later, and what we have seen is that vaccines and medicines can reach people faster without any compromise to their quality. One of the key principles for making this happen that we have applied within Moderna is the democratization of statistics. In order to move as fast as we needed to, we couldn't rely on a few statistical experts. We had to get statistical tools into the hands of scientists and engineers working on many fronts at once. And I'll give you a little detail on how we made that work. And then I'll finally also share an example from vaccine stability of where statistics and design of experiments are having a huge impact. So as I mentioned, vaccines have to be delivered and administered to have impact. So the same as a cancer treatment or a gene therapy, until the vaccine can be administered, it's not much use to the world. In the past less than a year, Moderna has produced more than 200, administered or distributed more than 200 million doses received by more than 110 million individuals in 45 countries. And it's very difficult for me to prepare this slide because the numbers change so quickly. We just keep going and going as fast as we can. But it's when shots get into arms that we really see the impact of the vaccine. This is a huge change, and these taking some of these timelines right out of the news, the mRNA 1273 sequence was finalized in January of 2020, and just 63 days later, the first participant was dosed in the phase 1 study led by the NIH. And then less than a year later, we had emergency use authorization from the FDA. a huge part of our ability to accelerate is due to the participants in that COVE study, the phase 3 study. One thing that Moderna is very proud of, we're all very proud of, is that as the study was enrolling, at a certain point, we realized that some of the most impacted groups were not fully represented in that study. So we actually slowed down enrollment to ensure that groups that were most impacted by COVID were well represented in the study. And we are extremely grateful to the participants who stepped up to participate in that and have such an impact. Traditional drug development averaged 12 years. There are, different estimates out there. This is from a 2015 publication. And for those of you who don't work in pharmaceuticals, this may be pretty surprising, but what happens is we discover a drug and then it begins to proceed through preclinical development, getting it ready in the formulation or the image that it can be used. Ultimately, we start clinical trials, phase 1, phase 2, phase 3, all very well defined with quite well established requirements. And in traditional development, along the way, in parallel, we do analytical method development, qualification, and validation. And then in what now seems like a leisurely pace, then once we have analytical methods ready, we would begin full-scale process development. And part of that that's very important is to do stability studies where we demonstrate the shelf life of the product we're working on. There is some work done in parallel, but much of this in traditional development was done sequentially, a very logical order. And a big part of the reason for this to be sequential is to minimize the risk to the manufacturer, because you can imagine if a product failed in phase 1, we wouldn't want to invest the resources to continue it through phase 2, phase 3, and so forth. And finally, we have health authority review before the product can be approved and distributed. So this is the typical traditional approach. What we do with accelerated development, so beginning with oncologic products that were breakthrough designation like Keytruda, then into gene therapies for unmet medical needs with terrible genetic illnesses that were on a fast track to impact people's lives, and a product was needed very quickly. And then most recently for the COVID vaccines and now treatments as well. What happens is anything that could be compressed or done in parallel was done. So clinical trials, the phase 1, 2, and 3, there was some combining and overlap depending on the product. The health authority review also began, they call these rolling submissions, where the review begins before everything is ready. But the health authorities like FDA and European Medicines Agency, they realize that there are people quite desperate for these medicines or vaccines, and they can't wait. So this is done in a much shorter timeline. Of course, as I mentioned, part of the reason we could accelerate the COVE trial was the willingness of participants to enroll very quickly. What we also have to do is make a lot of these activities in parallel. So analytical method validation, process development, it's common now to have process development well underway before the analytical methods are completely finalized. So if you've been in Doug's courses for a while now, you probably are recognizing that this presents a great opportunity for design of experiments. So to have a methodology that allows you to bring structure and organization to the experiments, both on the analytical and process side, it's a great advantage. We can see from the beginning what needs to be done, and then we fill in all the pieces as quickly as we can with much of that work going on in parallel. And Janet Woodcock from the FDA has been speaking about this for years now. The breakthrough designation program from the FDA, as well as similar programs from other agencies, that really put the spotlight on that the clinical trials were no longer the rate limiting step. And the biggest challenge shifted to become ensuring quality manufacturing and access to diagnostics. So it's a huge change in emphasis. One of the outcomes of this has been that in the past, you would hear a lot about clinical statistics, and of course we still do, but this shift towards accelerating the manufacturing development and distribution has put a lot more emphasis on the statistics for manufacturing support. And I know I'll slip into this jargon anyway, so I'll explain that in the pharmaceutical industry, we call the support for manufacturing and development CMC, which stands for chemistry, manufacturing, and controls. And that's why in my job title, it says CMC, statistics and modeling. And CMC actually comes from the section of these license applications that describes all the chemistry, manufacturing, and controls. And statisticians nowadays have a huge input into those sections. I spend a lot of my time authoring, reviewing, and updating sections for describing those control systems. So this shift, I think what we often hear about is the sexy part, which is the discovery. So molecular biology, artificial intelligence, genetic sequencing, precision medicine, these are all great breakthroughs, very exciting. What we don't often see the headlines about is what it takes to produce a medicine or a vaccine at high volumes, affordably, to scale that up, to scale up both the manufacturing and the testing. So what we do is use quality by design as a roadmap. I think of it almost like an acceleration ramp. So it's that structure, that approach, which is heavily dependent on statistical methods, including DOE, that gives us a framework for how do we get from these unbelievably great discoveries to high volume, affordable, and scale up and distribution. So in order to make that work at Moderna, one of the keys to this success has been establishing a community of practice for statistics. And I think of this as the democratization and comprehensive diffusion of scientific method. There's George Box used to talk about this, and he wrote some about it in 1995. So we have developed a group of scientists and engineers who share best practices, train each other, and work on building this community of statistics application. This is a formal initiative in technical development at Moderna, where we have sponsorship and have defined the various objectives for, including things like identifying statistics champions in each functional area, providing training for them, having a recurring community of practice workshop. And I have to say, whenever I am able to attend one of those, it is delightful, because what I'm seeing these talented engineers and scientists doing is really eating up design of experiments, applying it to their work, and coming up with very interesting applications. I think it's true that some of the great innovations in statistics happen when it's in the hands of the practitioners, and not just a bunch of statisticians, proving theorems and coming up with new distributions. I mean, it's really been fabulous to see what can be done with people who are so enthusiastic about applying statistical methods. We have representatives from most departments within tech development, with a wide range from brand new employees. We have many brand new employees as we're growing so fast, but actually a a really strong representation from people who've worked at Moderna for 4 years or more. So it's a great mix of people and just a wonderful collaboration. Part of the setting up of this community of practice was to do a simple survey about statistical things, which is statistical types of work which people might need to do. And almost everybody does some form of data generation or analysis and reporting. Many also do a lot of experimental design and modeling. So experiments are getting designed whether or not statistical design of experiments techniques are employed. So what we're doing here is encouraging adoption of DOE really broadly within tech development. The philosophy is that if an engineer, a scientist comes to a statistician, gets advice, walks through just setting up a single DOE, that's fine for the moment. But really, for ongoing development, what we want is to teach people to fish so that they can come back over and over again, learn and apply DOE to many different problems. Trying to think about what could be, what statistical methods can we teach people to fish versus automate? It's kind of tricky because in biotech and pharma, we actually use a lot of statistical methods. And the way I've been thinking about this is on the vertical axis, we have the volume. So there are some statistical techniques we use at a very high volume, maybe repeated analyses where we have multiple attributes or parameters, or we do frequent updates. And then at the lower end of the vertical scale, there are analyses we do once in a while, maybe even one and done, and they're much more specialized. From left to right, we have on the left end, low potential to standardize, and on the right end, a high potential to standardize, which is just the reverse of complexity. So very complex methods, of course, are tricky to standardize. So what we've done is carved out this space where we think it makes sense to teach people to fish. And we're really focusing there in the DOE community of practice. On this lower end, where we have high complexity and any sort of volume, we still do a lot of consulting, and we are expanding that as quickly as we can. Over to the right, we have methods that are so high volume and simple that we are looking to automate those and automating where we can. So for example, in our stability programs, we have setting release limits. Those are internal release limits for products. That is a really complex thing, but we only have to do it once in a while. So this is something where the statistics group is extremely deeply involved. On the other hand, there's stability modeling. We do ongoing stability shelf life testing all the time, and we're continuously getting new results. So that's something that people in analytical development and process development teams can certainly learn and take on. And shelf life estimation actually can be nearly automated. It still requires a careful review of the data and somebody to assess that the models are adequate, but much of that is pretty well defined. Finally, way over on the right, we have stability monitoring for out of trend results. At the point where we've established what the expected stability profiles look like for our products, we also want to just monitor new results over time to make sure nothing is inconsistent with our expectations. That can really be automated. So this is how we're thinking about what we can teach people to fish. And this is some more of the methods that we use routinely. I won't go through every one, and this isn't even complete, but there are a lot of statistical methods with a lot of opportunity to teach people to fish in that center ground. I'm going to talk a little more in depth about what we've done to teach people about DOE and how to do it themselves. So we certainly maintain this core set of design of experiments where we need to be somewhat innovative. Things like mixture designs are often challenging to set up. We have many experiments where we have constraints on randomization, or we have we're actually doing a lot where we're expanding on original sets of experiments and adding in new runs with different factors. So those things we tend to keep the statistics experts involved in. But here, for process characterization, design of experiments, and a lot of the multivariate modeling we do, we are putting that in the hands of the practitioners. Part of what we're doing is setting up standard workflows. This should look pretty familiar to you, I'm sure. There's nothing earth-shaking here. This is just always start with the problem statement, a clearly written one that is actually written down, and a diagram of the process or method you're working on. And then you step through with things which should be familiar, like define what are the responses and factors. What sort of model form do you think will fit? From there, you come up with a proposed design. Most important part, run the study, analyze the results, and then all of that provides the output that is needed in our control strategy. We have to define a control strategy for each set of unit operations that are important for vaccine manufacturing, and it comes right out of these DOEs. This is just a simple example of the kind of block diagram that I encourage people to always be able to write down. We have a lot of jargon in the pharmaceutical industry, so CPPs are critical process parameters or factors in a design. CQAs are the critical quality attributes or responses. And by sketching out the unit operation, we can also have a good discussion about, are any of these difficult to control at full scale? Are any of them hard to change during the study or during actual operation? And after running the study, fitting the model, it leads us straight into being able to do things like prediction and Monte Carlo simulation. So a graph like this might be included in our control strategy document. And these days, I don't even see most of these. Our scientists and engineers are producing them and becoming very competent in using this sort of output to define normal operating ranges and proven acceptable ranges. So this statistical toolbox that is so key to quality by design is enabling accelerated development. One of the most interesting aspects, though, is shelf life. So we're all familiar with shelf life, even for a loaf of bread. You can think about if you have a loaf of bread and you put it in the freezer, you can keep it edible for longer than if you put it in the refrigerator, or if you leave it at room temperature, or if you leave it in a hot car. So we're all familiar with that concept that storage, and especially colder storage, helps us maintain a longer useful life for many products we're familiar with. This is the same for vaccines. Colder storage generally will help us maintain a longer useful shelf life. What we have here are, back to this illustration of how we've compressed the timelines. In this super compressed development that we are working with, our shelf life claim may even be longer than the critical path. For the mRNA 1273 Moderna's COVID vaccine, the shelf life claim is 9 months. It was only 11 months from the beginning of the finalization of the sequence to the emergency use. So you can imagine a lot of this work had to be done before we could complete full-length real-time stability studies. So we had to come up with some other ways to support our stability shelf life claims, and those relied heavily on statistical methods as well. So I shouldn't say just statistical methods, though. I mean, really, our ability to model and predict shelf life is based on our very strong understanding of the science that is the basis for mRNA vaccines. So the lipid nanoparticles that contain the messenger RNA, these are highly engineered and well characterized, and there has been many years of work going into these. We do characterization at many levels, and so the science is really strong. And we have a very good concept of the mechanism, the primary mechanisms for degradation. The messenger RNA itself is represented here, and we know that integrity of that RNA chain is essential for the product to be active. So when we talk about messenger RNA, its function is really to translate, and it can only translate if it has this full-length intact chain. So at one end, we need a cap, and at the other end, we need a tail. If the chain breaks at any point along the strand, then the RNA becomes inactive. It does not create safety risks because our bodies are already full of RNA, and these are just fragments of RNA that become inactive. They're metabolized the same way as endogenous RNA, but it's really important for product activity that we understand the proportion of RNA that remains active. And as you can imagine, these RNA chains tend to break over time, and at higher temperatures, they tend to break at a faster rate. This is a snapshot of some of our data at three of the key temperatures, the frozen temperature, refrigerated, and room temperature. And you get the sense that this was early data, actually. We have much more data at this point. But even with our early data, we could see that the degradation was consistent and predictable. And more than that, it lined up with our understanding of the mechanism. So it follows first order kinetics, as you'd expect, and very consistent for lots that were manufactured at different scales. So part of our phase 3 clinical trial was we tracked every single dose from the point of manufacture all the way through to the point of administration. And from that, we knew for every single dose, how much time did it spend in the freezer, how much time refrigerated, and then finally at room temperature before administration. And the graph on the left shows for the 15,000 doses at day 1, and then the 15,000 doses for the second dose at day 29, we have a very good estimate of this intact RNA for every single dose. This was done based on our stability storage studies and using Monte Carlo simulation. We know what the starting intact RNA was for each of the lots used in the trial, and based on how long it's spent at those other temperatures, we can forecast that. When we look then at the early breakthrough cases, we were able to show that the gray mass of points represents the distribution of this intact RNA for all 15,000 doses for each day, each dose. The red asterisks on the right represent the estimated intact RNA for the 11 participants who had those first breakthrough infections. And what we see here is just no relationship between this intact RNA within the range we have defined by our minimum specification and the breakthrough infection. And what I should also mention is that we, in this phase 3 trial with this range of intact RNA, these are the doses that produced 94% efficacy. So we have demonstrated in clinical studies that the range of this intact RNA, as long as it's above this lower limit, is able to produce this 94% efficacy. So what really matters to the patient is what's in the medicine or vaccine at the time they receive it, not at the time of manufacture. So the way we set specifications for this, it's pretty interesting. We start with this clinical minimum for intact RNA that we have established through the phase 3 trial, as well as some other evidence I'm not showing here today. But we have an understanding of what is the minimum below which we should just discard the vaccine. And above that, we use our stability studies to build in expected losses for room temperature use. So we start in the lower right corner and work our way up. We build in the expected losses at room temperature, during 5C refrigerated storage, and then during -20 frozen storage. And then we add to that a buffer that accounts for the uncertainties we might have in things like every time we measure this critical quality attribute, there's some assay variability associated with that. So instead of setting specifications based on our process capability, we set the specification based on our clinical minimum, as well as our predictive models for the losses during shelf life. And then we compare our process capability to that specification to make sure that we are, all our manufacturing sites are capable of meeting that. This ended up being very important during the pandemic. As you can probably recall, in the early days, we were just making the vaccine as fast as we possibly, well, we're still making the vaccine as fast as we could. And it was getting distributed very quickly. So the trade-offs we can make between frozen storage, refrigerated storage, and room temperature, those came in very handy in the very early days. As the vaccine distribution has evolved, we can continue to make trade-offs and do things like adjust our minimum release limit to give greater shelf life, for example. So we are now at a point where we're in the hundreds of millions of doses from a year ago, when we hadn't even received emergency use for this authorization for the product. And statistics played a huge role in this, both in the design of experiments for process characterization, our setting up of shelf life claims, and in many, many other aspects as well. So this has been such an unusual time for all of us. And I've been very fortunate. This is my mother a year ago, well, less than a year ago, January 4th of 2021 was when she got her first dose. It happened to be Moderna, which made me very happy. And she's scheduled to get her booster maybe today, now that I think of it. So, so this is, it's something that has impacted the whole world, COVID, and trying to get vaccines out to as many people as we can. But it's also been very personal. So it's just been a fascinating journey to be part of. And to summarize, we have moved into a whole new era for vaccines compared to what we worked on decades ago with the very first vaccines. It was much less well characterized. We're now in a place of precision vaccinology. So when we understand a genetic sequence, we can very rapidly develop a vaccine targeted specifically to that. The journey from discovery to large-scale distribution has depended a lot on the democratization of statistics, particularly design of experiments. And I hope you found the vaccine shelf life example interesting too. So just never forgetting that the reason we do all this is because of the people who need these products. So I always think of Connie as we're doing this work. And the same approaches apply certainly to degenerative genetic diseases and now to infectious outbreaks. It's this tying together of science and statistics for human health. It's the George Box would say the catalysis of the scientific method itself. So thank you very much for your attention, and I'll be glad to chat with you, Doug, about any issues this may have prompted.
Julia, thank you. Thank you very much for this presentation. I found this really quite interesting, and I also want to thank you for your comments about Connie. Connie was my PhD student, and she ended up her career, of course, teaching here at ASU, and she did her dissertation work on robust design, which is how we all got connected. And miss her a lot. She's somebody that I really became a very good friend of, and she left us way too soon.
Yes.
A couple of things in your talk that I found really interesting. You have a sort of a step-by-step process of problem solving that I'll remind people in the course that we covered something very much like that in the early part of our course. And you also show a block diagram that's very similar to one that I also show. And I think those kinds of concepts are really important for people to understand as they apply statistical tools in general, but experimental design specifically. Okay, we have some questions, and maybe we ought to try to get to those. And
sure, and I'll just say that I'm sure that in putting together that step-by-step checklist, I was consulting your books on design of experiments.
Oh, well, that's okay. You've got some stuff there that I don't, which is good. It's good. I hope people take ideas and expand them. You may be familiar with the article that David Coleman and I wrote in Technometrics on planning for a designed industrial experiment. And I've run into dozens and dozens of people that have said that article really sort of gave them a framework for figuring out how to get experimental design methods used properly in their applications. And that's great. That's what you hope happens. That's why you work on that.
That's right.
So here's a question, and I'm interested in your input on this too, Julia, but let me read the question. It says, I'm working on engineering education, and I want to find a function between student satisfaction, which is my response, and I can measure it through different models, and I have different factors. It can limit to 5. The problem is I can't do experiments, and I have only data from, he says, previous experiment, but my guess is that this is not experimental data, but it's more happenstance data. This is what you get. Can I consider every data like an experiment? And if not possible, how can I find a mathematical function between those factors and my response? I want to apply design of experiments in my study, and which part can I apply on data and not experiments? I kind of think that what he's looking at is a regression problem. Don't you think?
Yes.
I've seen studies like this in which you try to predict not satisfaction necessarily, but performance. What does their grade point average at the end of the first academic year look like as a function of a lot of different things that you can measure or that you have prior information on, scores on standardized tests, performance in secondary school, experience, strength of recommendation letters. There's lots of stuff and I suspect that that's maybe the kind of thing he's looking at, don't you?
Yes, and I actually see something similar even with engineering and chemical data, is often before designing their first real statistically designed experiments, people will have a bunch of data that they happen to already have.
Right.
And I do encourage them to see what they can learn from that data. Of course, the big caution is that if you don't intentionally change the variables or the factors, you can never be sure you're seeing cause and effect. It might be a coincidence.
Yeah. Big concern.
Oh, yeah, yeah. It shouldn't be the end of the story. But on the other hand, a lot of times we can learn quite a bit about maybe even surprising factors.
You sure can.
Yeah. And I'm a big fan of some of the exploratory tools, like just doing clustering, or I actually like random forest quite a bit. Just see what pops up as maybe having an impact.
I think that's a great idea. Of course, the other problem with using that kind of data is sometimes the important things never got collected. I think we've all seen that. The really important factor, nobody ever thought about it, and it never got collected. So,
exactly.
but you can learn so much from looking at this kind of I think of it as historical data.
Yep.
It can be really
important. We actually have that problem, and even worse in a lot of manufacturing data, because we have intentionally prevented the most influential factors from varying too much. So then we go to look and see, is there any sort of predictive model? But we have, by design, kept the most important factors within a tight range. So we will never find them in a model of historical data.
I had that experience with a consulting client a number of years ago, and they were a chemical company, and we wanted to, the engineering guys wanted to run this really nice characterization experiment, and the bosses didn't want to let that happen, because they said, oh, well, you don't need to do that. We have all this historical data. And so we analyzed the historical data, and ended up in being involved in a presentation. And one of the results of this was, well, this factor temperature is just not important, and some of the engineering people just started laughing, and when the engineers are laughing at your results, you're probably wrong. They said, well, we know that that's important. That's why we control it so closely. They actually had a PID controller on this temperature variable, and it never changed. So yeah, you just have to be careful when you look at that kind of data, because it can be a problem. So we got another question from Zoom chat. Curious how animal-based testing, ethical of course, contributed to our fast realization of the vaccine, and how that animal testing longer term is helping us know more about durability of the antibody response. Less of a concern about animal testing, and more of a how are we staying ahead of the curve on studying durability. So I don't know, I guess he's looking for some comments about animal testing in things like vaccine development.
Yes, and certainly one of the satisfactions of working as a statistician is we get to help minimize the number of animals who are sacrificed for this kind of testing. But animal testing does remain very important for understanding things like dose response behavior. And in many cases, we do have good animal models that allow us to get a big head start. We always have to follow up with testing in humans in the end, but to confirm things. But a lot of our design of the product does use quite a bit of animal testing.
I continue to be amazed at how quickly the COVID vaccine was developed, because I know a little bit of history about polio vaccines, and people talk about the great flu pandemic of 1918. Well, there was also a widespread polio epidemics in that same time era. And it wasn't until somewhere around 1933, 1934, 1935, that the first polio vaccines actually appeared and they didn't work. They were not successful. And then Jonas Salk worked on it for years until he finally got something that worked. And that was in the early and mid 1950s, because I was one of the participants in Salk vaccine trials as a kid.
Wow.
I grew up in the 50s, and so I think what's been done with the COVID vaccine development is remarkable science. And it's great to see that experimental design and statistical methods have played such an important role in that.
Yeah, and I mean, one of the things that I shouldn't fail to mention is that there was a solid 10 years of development at Moderna, as well as development in lots of other places for the RNA approach to vaccines. And I consulted, I joined as an employee about a year ago, a little over a year ago, but I consulted with Moderna for about 4 or 5 years before that. And they had a vaccine candidate that was quite well advanced at the time that COVID disrupted all our lives. So it was not too difficult to shift focus to attacking a different virus, but with many of the vaccine platform features that had been designed over 10 years and more. So it's kind of deceptive. I mean, we didn't know exactly what the virus sequence would be until that January, but we actually had been learning and designing a lot about how to create an effective vaccine using mRNA for a good 10 years before that.
Right. So here's a question from the Zoom chat that's directed actually to you. So let me read it. It says, do you think there's more opportunities for design of experiments in pharma compared to more traditional commodity chemical industries?
I'd have to probably say yes. I think I've spent my career moving from specialty industries that had a lot of fascinating technology. As those matured and became commodity, I've been able to learn and move with those changes. So my very first job was in a paper mill, and then 17 years working with specialty chemicals, and then into viral vaccines at Merck for 10 years, and now mRNA vaccines. So I do think that the opportunities to do interesting experiments, they tend to track with the advances in science. Although design of experiments, of course, are extremely valuable for things like optimization of current processes. So they can be useful just about anywhere. But I find them most interesting, I guess, in some of the more advanced scientific areas.
I've always felt that experimental design is a broad tool that you can use all the way from the very conceptual thinking about a product or even a service, and moving that forward through development and into manufacturing. And if you're not integrating this into those product realization activities in your business, you are leaving money on the table.
Yes.
And you're not developing products as quickly as you should. And if somebody is using this methodology and developing competing products and they beat you to the market, you could be out of business. And so I just see this as such a vital tool that needs to be much more, broadly embedded in a lot of industry. Okay, another one for you. Can you explain vaccine shelf life experiment very precisely once again?
Sure. So the experiments that give us the foundation for shelf life are actually very simple. We take representative samples of the vaccine material. We do this for the final product, and we also do this for the materials that go into making that final product, like the bulk material before it goes into vials. We take those and we put them in controlled temperature storage at the temperatures that are of most interest for us. So for example, -20 is a representative temperature for the frozen storage of our vaccine. 5 degrees C is a representative temperature for refrigerated storage, and then 25 C is used to represent room temperature. And what we do is we test the sample when they go into storage at the very beginning, and then we test them at predefined time intervals following storage at those temperatures. So for example, the -20 C study, we might start at the initial time 0, and then we pull a sample at 3 months and test it according to a whole variety of test methods. We pull another sample at 6 months, test again, and another at 9 months and 12 months. And depending on the product, we might go out much longer than that. It really depends on how much shelf life the product needs. And we do that for multiple lots. So we make sure that our samples represent different manufactured lots. If there are different scales of manufacturing or locations, those are also represented. So we end up with a bunch of studies that show us, is the loss we are expecting to see at a specified temperature, is that consistent lot to lot, and is it predictable? And very importantly, does it match our scientific expectations for the rate of loss? Is it, a straight line, or does it match first order kinetics, for example? So the studies are very simple, and the analysis is somewhat predefined, but like any other real data, there are always twists and turns, and we have to use look at the data carefully before analyzing and all of that. But then at the end, from each of those studies at different temperatures, we can estimate a loss rate and an uncertainty around that loss rate. And then we plug those estimated loss rates into an overall model, which actually is in a World Health Organization guideline. And we're taking the best of the old and the best of the new. We're using this very advanced characterization for mRNA vaccines, but we're using a really well-established model from the WHO to combine all of that information and come up with a shelf life.
Great. Another one for you. Do you foresee that the accelerated drug development, analytical method development, process development happening in parallel model will prevail? Or do you think the industry will return to the traditional sequential model for most of the products? And related to my previous question, do you think many companies will also try to implement a democratization of statistics?
Yeah, so I'd say I'll start with the second question. I think that many companies would like to implement democratization of statistics, partly because it's so hard to find statisticians who are qualified to work in CMC. It's really, we are recruiting, and it's always tough to find people. For me, the ideal candidate is someone with a solid background in science or engineering who develops an interest in statistics and maybe goes on to get a master's or a PhD. Those are wonderful candidates for this type of work. But because they're so scarce, I think that companies will be eager to democratize statistics as much as possible. In terms of the accelerating versus traditional model, I think that that it is being done for many products, but selectively, really based on the need. Because accelerating a product to this extent requires tremendous focus and resources. One of the most challenging parts of this has been people I've met who work for health authorities, the reviewers and the inspectors. They have been handling such a high volume of work these last couple of years. It's not something that would be sustainable for every potential product out there. It's got to be done somewhat selectively. And then also the speed of the clinical trial, that isn't always possible. In fact, we've seen the reverse during COVID. So for testing products that are aimed at COVID, preventing or treating COVID, we had the advantage of very high visibility, high impact. So it was relatively quick to recruit volunteers. And the prevalence of the disease allowed us to get results really quickly, unfortunately, because what you're looking for in those clinical trials is to see, do lots of people in the placebo group become infected? It's a very unfortunate outcome, but because that was happening so quickly, the trial could be done quite quickly. Of course, for many diseases, that won't be the case. Luckily, you know, it would be terrible if all diseases out there were acting that way. So I think that the accelerated model will become a standard for products with a very high unmet medical need. But I don't think it will be, you know, a one size fits all. It would be very difficult to implement that across the board.
Julie, we got one more question that we might be able to handle here before the end of time. And again, I think it's for you. According to your experience, is there any statistical software that you would recommend over others for experimental design and formulation of drug product for the pharmaceutical industry? And I was going to actually ask you that myself, so I'm glad that came up.
Well, this is kind of a fun debate within my growing team because people have joined my team with a whole variety of different backgrounds. So within our team, we use JUMP extensively. We use SAS. We also now are using R and Python and MATLAB. And you probably pick up the influence of bringing people in who start as scientists or engineers. They tend to like some of those tools. There's also for design of experiments, those are the main ones. I actually am also a longtime fan of Design Expert, particularly for mixture designs. So I'd say for me personally, I use primarily JUMP and occasionally Design Expert as well. But I'm also kind of agnostic about it. Like I really, if somebody can get the software to do what they need, that's great, as long as, you know, we're assured that it's high quality. There is an advantage though to people tending to use the same software, is it's very easy then to share scripts or code. Yeah. Or so that.
Yeah, I certainly agree with that. That's you know, some commonality is always good, and I'm always nervous when people start writing their own computer code. That sends shivers of fear running through me, because I know how many mistakes that I've made over the years writing computer code. Yeah. Okay, so we are at the end of time. Julie, I want to thank you so much for your insights. There were some questions we weren't able to get to. I think there are 2 or 3 that are remaining. We'll review those and get responses directly to you. Julie, I'd like to probably run my answers to those questions by you also to get some input. Our final Fireside Chat for 2021 is going to be on Wednesday, December 8th. And information about the topic and the guest and how to get involved in this will be in the Design of Experiments Coursera courses. So there's another one scheduled for this year, and then we'll resume with a full set of them in 2022. Julia, thank you once again. It's great to see you. And
yes, great to see you, and thank you so much for the invitation.
Oh, we need to do this more often.
Yes.
Bye-bye.
Bye-bye.